Hello, everyone. My name is Huda Alchikh Omar, and this semester I did undergraduate research with Dr. Kang at the University of Houston downtown. For our project, we looked at the role of Drosophila UNC13, alcohol-induced hyperactivity and sensitivity. Alcoholism is one of the most important psychological problems today. And to study how ethanol changes neural activity and how that reflects on behavior, we used Drosophila melanogaster, which is one of the genetic model systems used to study genetic mechanisms underlying alcoholism. Ethanol has a big effect on the presynaptic activity in neurons. And one presynaptic protein that binds ethanol is Drosophila UNC13. UNC13 is a conserved protein that is found in both humans and flies. UNC13 is an active zone protein that is essential for synaptic vesicle fusion. UNC13 binds diacylglycerol, which facilitates the vesicle fusion. When ethanol is present, it binds UNC13 and inhibits the binding of the diacylglycerol. And this results in ethanol-induced responses like alcohol-induced hyperactivity. It happens when the flies are first exposed to ethanol, and this behavior is also reflected in humans. After a period of time, when ethanol concentration has accumulated in the body, we have sedation. In our research, we tested if UNC13 mutant is less responsive to alcohol-induced responses. To test our research questions, we used two assays. The top one here is called fly bar loss of writing assay. For this assay, we used it to measure the sedative effects of ethanol. We first load the flies into these 4 tubes, and we can control the water and ethanol concentrations using the air pump and the flow meter. In this assay, we measured the time half of the flies lost their writing. And this assay over here is called the fly gram assay, which stands for the fly group activity monitor assay. We first clean the arena here, and then we load the flies, and we make sure that they are first only exposed to water. We let the flies rest for a little bit of time, and then we run the program. The first 5 minutes the flies are only exposed to water, and then we turn on the ethanol for 10 minutes. After that, we turn off the ethanol and we run the program for an additional 5 minutes. This assay was used to measure the hyperactivity of the flies when exposed to ethanol. For both of these assays, we used male and female flies, the UNC13 and the control flies, and the flies were 2 to 5 days old. After analyzing the data, we found that the UNC13 mutant flies are less sensitive to ethanol sedation effects. The UNC13 males and females were less sensitive, and they took longer to lose their writing reflex. The UNC13 flies have higher alcohol-induced hyperactivity. We chose not to show the error bars here because these graphs represents the mean of the active flies. And this part over here, it represents the time the flies were exposed to ethanol. UNC13 mutant flies showed more resistance to alcohol sedation in males and females, consistent with the UNC13's role in the synaptic vesicle release. Our preliminary data suggested that the alcohol-induced hyperactivity was also affected in these UNC13 flies. Some factors that we believe affected our experimental outcomes are ethanol concentration and unequal sample sizes. To fix this problem, we are going to test the flies using different ethanol concentrations by noting what does the ethanol concentrations do to the behavior of the flies and use equal sample sizes. Thank you for listening and have a great day.